Hi everyone, good evening. Welcome to our last IGERT winter seminar. Today we will be listening to a talk from Curtis Deutsch. Curtis is an associate professor in the School of Oceanography right here at the University of Washington. His lab is, or his work is focused on understanding the interactions between climate and the ecosystem, and he's focused a lot on biogeochemical cycles in the ocean. Curtis received his PhD from the Department of Atmospheric and Ocean Sciences at Princeton University and has a Bachelor of Science in Physics from Oberlin University. And today he'll be giving his talk entitled Climate Change and the Loss of the Marine Aerobic Habitat. So, Curtis.
Thank you very much. Thanks to those who've made it to the very last seminar. Despite the fact that the advertisements all had my title as a three-letter title, my shortest ever TBA, I filled it in at the last moment and you made it anyway, so thanks for coming. I have actually discussed most of this material on campus before this, earlier this year, so for those who've already seen it, I will forgive you for leaving now, but I should warn you that you won't get beer if you do. So let me just jump right in with the fact that most of you probably already know that the ocean is undergoing three fairly well understood perturbations, and two of them are illustrated here. The first fairly obvious one is that as the Earth's climate warms, the ocean warms. In fact, most of the warming of Earth's climate is going into the oceans. And we can sort of see that in the historical record, but we can also use model simulations to project how strong that effect might be going forward. This just shows you a compilation of a wide variety of Earth system models used as part of the most recent climate assessment by the IPCC. And you can see that there's some range of uncertainty associated with just the structure of different models, but we can project it on a global average over the upper 500 meters of the ocean. We're looking at something like a couple degrees warming over this century. Now, associated with that is a drop in the oxygen content of the upper ocean, in fact, parts of the deep ocean as well. And that also has some range of uncertainty, but if you take a global average and leave aside the rather more difficult large-scale pattern of that oxygen loss, you find a fairly robust trend toward something like 10 micromoles per liter of oxygen declining over this century. And that's, against a background oxygen content in the ocean that equals something like a couple hundred. So it's not a huge perturbation, but it can be significant. The third, which you may or may not have guessed, is the declining pH of the ocean. And just for completeness, I'll state it, but I'm not going to talk about it, so I don't show it here. The prediction now is that some combination of this warming and loss of oxygen should pose problems for oxygen-breathing animals in the ocean by compressing the geographic space in which they have a viable habitat to live. And I really want to spend the next 40 minutes of your lives going into some thoughts on how that might work and how well we can know that it will work. So just to begin with, the idea that oxygen in the ocean has a big impact on the viability of habitats for marine animals is probably not very surprising, and we have plenty of fairly recent evidence just re-illustrating that point. One of the more dramatic forms of evidence comes from just south of here off the Oregon coast, which in around the early 2000s began to experience fairly dramatic perturbations to the marine ecosystem that were attributed to the loss of oxygen, especially in benthic communities on the continental shelf. So here's just an undersea image from August of 2000, where you see a fairly healthy assortment of fish on the seafloor, and then a little less than 2 years later, after a fairly severe kind of month-long or so hypoxic event, low oxygen event, you see the remnants of species of organisms that were killed off by that, along with lots of other worms and crabs and things. So this is kind of a very short-term, what we might call event scale example of how the loss of oxygen can wreak havoc with marine species. We get sort of longer-term perspective from longer time series. This one also in the California Current, but off Southern California, in which people have been measuring a wide variety of hydrographic and biological properties for several decades now. So going back to something like the late 1940s, 1949, I believe, it's one of the longest running measurement programs that includes oxygen anywhere in the ocean. And what you see when you correlate oxygen against the abundance of fish, eggs and larvae, for example, is a fairly strong correlation. That is, when oxygen is high, fish biomass tends to be relatively high. And furthermore, the variations in both properties have a very pronounced decadal kind of pattern to them, in which oxygen was relatively low in the early part of Cal Coffee measurement program. It increased into the 1970s and 80s and has subsequently declined in the recent couple of decades. So there's let's say, a circumstantial evidence for oxygen having a very important control on the abundance of fish biomass in the Southern California Current. The fish, by the way, are plotted as this strange statistical object, the blue line, but really the message you can get from this plot here. And then finally, just because I'm kind of fascinated by it in recent days, I had a very long-term perspective. I think it's actually kind of important to keep in mind, because as a lot of people work on the geologic past, they seek kind of past analogs for where we might be headed in a rapidly warming ocean in the future. And this one is holds a special place in that list because it's the most dramatic mass extinction event in the history of Earth, the end of the Permian, going into the Triassic. So here is a whole bunch of information that I just want to use to give you an impressionistic sense. Here on the y-axis is a time scale. If you can see it in very faint numbers in millions of years ago, so we're at 253 down to 249 million years ago, going from the Paleocene to the Triassic. And one of the key observations about that period is that, well, first of all, the number of species declined really dramatically, something like 90%, give or take, of all known marine species that were existing in the Permian basically disappeared by the end of that period and into the early Triassic. So in a whole bunch of different taxonomic groups, there was a rapid contraction of marine biodiversity. And there are a variety of different plausible mechanisms for that, but one of them is shown here, and that's these are isotopic indicators of the temperature of the seawater in which skeletal material was formed, in this case for conodont species. And you see that, well, there's a fairly simple conversion of this isotopic scale into a temperature scale, and it equates to something like an 8 to 10 degree warming of upper ocean temperatures across this time period of something like 1 million years. Right? So this is a big temperature perturbation, but it's also relatively slow compared to what we were looking at before. And notably, I don't think you can quite see it on this version, but somewhere right around here is a turquoise bar that shows you where the range of modern sea surface temperatures are. So this is not, this started from temperatures that are a bit cooler than today and went to temperatures that are a bit warmer than today, the total range being 8 to 10, again, degrees. So, I'm not going to comment on whether this is a reasonable analog for a plausible future, but it is important to keep in mind that the paleo record holds some fairly ominous warnings for us about the effect of rising temperatures and, in this case, declining oxygen in the ocean. Now, it's also interesting because it's one of the, relative to the earlier two cases, it's the only one where we have both of those phenomena happening at the same time. We have warming and we have oxygen loss in the late Permian extinction, unlike the two earlier shorter-term fluctuations where oxygen was declining but temperature wasn't really playing a main factor. And the reason why it's interesting to think about the two things operating together is that the understanding of fish physiology has led to the hypothesis that the two things are really kind of almost interchangeable. And the first indication of that, at least that got a lot of popular attention, was from studies on the common eel pout by Hans Otto Portner, in which the eel pout was grown at a variety of temperatures, temperature on the x-axis here, and just the simple daily growth rate of the organisms are plotted on the y-axis. And you can see, not unlike all kinds of organisms on land and in the sea, there is some kind of thermal optimum here with fairly rapid decline in growth rate as you get toward temperatures warmer than that optimal one. And the main argument put forward in this paper is that this decline in growth rate is basically dictated by a squeezing effect on the aerobic scope, which is jargon for the moment, but I'll come back to in a moment, that the fish faces. So here is the oxygen concentration at which these eel pouts, little kind of eel-like things, began to build up anaerobic metabolites in the body tissue. So as the temperature warmed up, they began to accumulate those anaerobic metabolites at higher and higher oxygen levels. So their tolerance for low oxygen was declining, one could say. And at the same time, just sketched here is a curve that will be familiar to all the oceanographers in the room. It's the solubility of oxygen as temperature increases. So his reasoning was that as the ocean warms up, the oxygen tolerance, the tolerance for low oxygen will decline, and at the same time, the oxygen availability in the seawater will also decline. Now, it probably is true that he's underestimated the slope of that black curve, and we'll come to that in a little while. But suffice to say that it indicates that that one can either generate a kind of low oxygen stress in an organism, either by reducing oxygen or by just warming the water. So the two things are sort of interchangeable at some level from the perspective of an individual organism. Okay, so here's what I want to do for the next 30 minutes. I want to pose this fairly simple question. Does this kind of picture of temperature-dependent hypoxia actually constrain the contemporary habitats of marine species? Can we tell that something like what's happening in that figure is actually happening in the real environments where species are living, not just in a laboratory tank with fish brought into it? And if we can use those ideas to understand where habitats are in our contemporary ocean, can we use that information then to make more mechanistic kinds of estimates for how habitat will shrink as the ocean warms up? And you can probably guess that the answer to the first question would be yes. And to do that, I want to try to weave together different types of data. All comes from different places. The first form of data that I want to show you is basically laboratory data, exactly of the type that you just saw from the figure put together by Hans Portner. And then I want to combine that with climate data to map what I'm going to call a metabolic index, which is an estimated ratio of oxygen supply to demand, and then match that against where the species for which we can calibrate a metabolic index actually live in the current ocean, and see what we can determine about how the combination of oxygen and temperature lines up with the revealed habitat, or the kind of realized niche, as some people call it, of these different fish species. Okay, so first a little bit of kind of, let's call it simple-minded scaling. What I'm aiming for is an index that measures the relative rates of oxygen supply to demand for a hypothetical organism. This is not a hypothetical organism. I think it's a fish, but I'm not a biologist. I just grabbed it off the web. And the point of it is to just show you that the water comes in bearing oxygen. It flows through some gills that have some area and some efficiency at extracting, transferring oxygen from the seawater into the tissue. And the rate at which the oxygen can be supplied into the tissue then is going to be proportional to something about the efficiency of that process, the oxygen molecules crossing from the fluid into across the membrane into the tissue, which we can think of as an exchange rate, probably per unit area of gill surface, for example. And that's going to need to be multiplied by some notion of the area of the surface over which the fluid and the organism are in contact and exchanging oxygen, which ought to scale in some fashion with the body mass of the organism. Okay? So body mass to some power delta, because we're unsure exactly how that scaling should look. And then it's ultimately driven by basically the partial pressure of oxygen in the fluid, assuming the partial pressure of oxygen in the blood is relatively small. This is the driving force for that exchange, pushing oxygen across the fluid organism barrier. Okay, so oxygen demand, we can sort of write down in similarly simple terms that the rate of oxygen that is needed to be consumed by the organism ultimately comes down to its metabolic rate, how quickly it's converting organic matter and oxygen into energy for biotic maintenance. And so here, of course, we're considering aerobic organisms, and there's a fairly well-defined relationship between oxygen consumption and the energy that's extracted in the various steps of respiration. But to simplify it, we can say that rate of oxygen consumption, the oxygen demand, is also going to be proportional to some kind of metabolic rate, which will vary widely among different species, at some kind of reference body size and temperature, but it will always go up with body size in some power law way, and people argue endlessly about what the exponent would be, and I'm not too concerned about that here, as you'll see in a moment. But as body size increases, metabolism increases. It probably does it sublinearly, but that's no matter for us. And then finally, metabolism, at least in a resting state, goes up as a kind of quasi-exponential function of temperature. So as temperatures go up, resting metabolic rates go up fairly sharply. And just to demonstrate that that this equation does a fairly good job of capturing observed patterns in resting metabolic rate across the domains of life. This is a compilation of such data, basically plotting alpha D here on the y-axis, the log of that against 1 over temperature. And there are variations among different large taxonomic groups, but they all basically follow this kind of exponential dependence on temperature with slightly different slopes and different intercepts, but we can work that all out later. Okay. So what I'm interested in, though, is the ratio of the oxygen supply to demand, because it's really when that ratio crosses below some threshold that you're going to start to see biological responses that could be rather stressful. So just going to simply divide those two expressions that I showed you before. Take supply divided by demand, and you get this this formula that I'm going to refer to with this symbol phi. I'm going to call it the metabolic index. It's just an index of supply to demand for oxygen. And you can see that the ratio of the two alphas comes out in this, we're just going to call it A0, so we're going to have to keep track of 2 letters instead of 1. The biomass gets raised to 2 exponents, the difference between those 2 exponents, one being the rate at which oxygen demand increases as size goes up, the other one being the rate at which the area for extracting oxygen from the environment goes up as size goes up. And then a third physiological parameter which governs basically the temperature sensitivity of resting state metabolism. So basically the slopes of those curves in the previous figure. And then there are 2 environmental parameters here. There's the partial pressure of oxygen in seawater and the temperature of the seawater. So this is a sort of cumbersome thing, but it has a very simple, intuitive meaning. It's cumbersome because it has a bunch of parameters that we might not know all that well, and a couple that we do know pretty well, actually, that you can just simply measure in the ocean anywhere you want. anytime you want, now that people have developed cool sensors for doing both of these things. Okay, so in any case, this ratio of oxygen supply to demand will depend on the physiology of the organism and the environment in which it's living. That's fairly clear. A more interesting interpretation of this thing, though, rather than being a supply to demand of oxygen for an organism in its environment, you can think of it in the same way as a measure of the maximum metabolic rate that an organism could sustain in that environment relative to its resting rate. Right? So if you're an organism where the supply and demand are exactly equal, then you've got a ratio of 1 here. The maximum amount of oxygen that you can get out of the environment just satisfies your demand in a resting state. If you were to come along and double the oxygen content of the water, then you could, in principle, do twice as much metabolism as you could before. Right? So this gives you a measure of the kind of maximum rate of metabolism that you can sustain. The problem is, again, that there's all these parameters that are messy and probably can't be derived from first principles, but we can get them from laboratory experiments. So here's the first use of laboratory data. We've just taken out of the literature a whole bunch of measurements of the oxygen concentration in a tank where specimens were kept in a lab somewhere, and the temperature of the water in that tank at which the organisms either began to undergo a switch into anaerobic metabolism or they began to die. And the two things are really kind of the same thing, just one takes longer. At least that's what we're going to assume. If you start to go into anaerobic metabolism, nothing changes, you're not going to last long. Okay? So those conditions essentially define what we could think of as a purely physiological threshold. That is, as soon as phi, this metabolic index, crosses below 1, you're in trouble, right? You can't even sustain the metabolism that you need in a resting state. Forget about going to try and find food or find a mate or things like that. So that's a purely physiological threshold, if you like, but that's what we have from laboratory experiments. And it turns out to be quite useful because if you set the metabolic index equal to 1 and you have measurements of this, of these two parameters, then you can actually identify the intercept, as the intercept in these curves gives you the parameter A0, and the slope of it gives you that E0, the temperature sensitivity. Okay? We also have to worry about mass, but we're going to come to that in a moment. In most of these cases, mass was held constant. And you see that for all these different species, there's a fairly conserved slope, that is a temperature sensitivity of resting state metabolism that gives us a fairly narrow range in this E0. The A0 values vary, and in fact, I've artificially inflated them here just so that you can see the different species clearly so they're not stacked up on one another. A0 is really not that important for our purposes. I'll come back to that in a moment. Okay. Now it turns out that temperature alone, you can basically account for over 80% of the variance in this critical oxygen value, the oxygen concentration at which species went anaerobic or began to die. Okay. Now we have a smaller subset of those species for which we actually can control for mass and size separately. And just to illustrate a couple of them, so we've got Atlantic cod here and sea bream here, body mass plotted on the x-axis, and this critical oxygen with that temperature normalization, the temperature term in there, which gives these weird units. And you see that in the species we looked at, basically the oxygen the tolerance for low oxygen decreases as the body mass increases. Okay, that is, a larger specimen of Atlantic cod will, according to this laboratory data, will die at a higher oxygen than a smaller organism will. Okay? And from that we can get, you can barely see it, the net exponent on B. So that's not the metabolic rate scaling versus size, which would probably be something like 3/4 or 2/3 or 1 or something. And it's not the allometric scaling for the rate of oxygen exchange, it's the difference between the two. So it's smaller than either one alone, most likely, And in fact, we get numbers from 0, in which the two things exactly compensate, all the way up to about 0.3, in which they're mostly compensating, but not exactly. And that's fairly commonly observed. A nice review of the question here in this paper. But for the species we have, basically smaller is a little bit better. Not dramatically better. And sometimes it's sort of indifferent. But it's none of the species we looked at is it worse to be smaller from the point of view of hypoxic tolerance. Okay. So now we've got all the parameters we need to estimate the supply-to-demand ratio, or if you want, the maximum sustainable metabolic rate in a given environment for these different species. But that really just tells us, first, what the physiological tolerance is. But of course, these things were growing in tanks, and in nature, they're going to need to consume a fair bit more energy than they would just sitting in a tank waiting to be fed. And so here we would like to identify more of an ecological threshold. That is, how much energy do you need to consume relative to your resting rate in an actual environment, right, where you're out doing stuff. And this has been measured for quite a long time, going back to the 70s in lots of terrestrial taxa. They basically inject isotopically labeled water in the animals and then collect them days later, and using kind of typical isotopic manipulations, figure out how much organismal respiration they did. And what they find is that for virtually all terrestrial taxonomic groups, the rate of daily energy expenditure relative to what they would do in a resting state, same animal, just same experiment, in fact, done on an animal kept in a cage, is something like 2 to 5 times, okay? A factor of about 2 to 5. So, in fact, all of us sitting in here do the same thing. On an average day, if you're not at sea, probably, you'll burn about 2 to 5 times as much calories as you would just sitting there. Okay? So what that tells us is that this phi of 1, which is a threshold for physiological survival, is really a kind of bare minimum. What you really need is something like a phi approaching these kinds of numbers. You'd better be able to get up, twice that much energy out of the environment. You better have enough oxygen to get twice as much energy as your resting state requires, maybe even 5 times as much, but somewhere in that range, in order to be sort of ecologically viable, okay, to have ecological survival. So the question is, what would those numbers look like for these marine organisms? So first we need to look at the distribution of this metabolic index, which now, given the parameters we get from the laboratory data, we can just map using climatological fields of oxygen and temperature. So I'm just going to show you a couple of these large global distributions before zooming in on where some of these species live. So this is global annual mean metabolic index over the top 200 meters. Probably, I guess, maybe most fish are in that range. Anyway, it's arbitrary depth bracket here, and it looks basically the same as you go deeper with some interesting nuances. In any case, the point is that I've normalized it to some tropical reference point, and the only reason for doing that is that that makes this pattern basically independent of which species you're talking about. Okay, so if you go back to the metabolic index, you got that big A0 sitting out front, and that's different for different species, and that's going to give this pattern a different absolute scale. But all the species have a very similar E0, which is their temperature sensitivity, and that E0 basically is the lever that controls how sensitive you are to temperature versus oxygen. Okay, and they're all fairly narrow in this activation energy E0. So basically, all the species will look like this once you normalize to some given place on Earth. I just took the tropics. So the tropics are 1 here on average. And then you can see that as you move toward the high latitudes, the metabolic index goes toward something well in excess of 10 times its tropical value. So what that tells us, and so the reason for that is fairly simple. That is, from the tropics to the high latitudes, you get 2 benefits as an organism. You get lower temperatures, so your resting metabolic rate goes down, and you get more oxygen, so you can actually more easily get energy out of the environment. And when you put those 2 things together, you get something like maybe a 20-fold variation in that potential ratio of oxygen supply to demand. Okay? So the other consequence of this is that if you think about a species that has some wide range, it's going to be most constrained by its aerobic environment at the southern, or let's say the equatorial range boundary, right? It's going to be, if it's constrained at all by oxygen and temperature, it's going to be more in the tropics and not in the high latitudes. Okay? So just a quick comment on the vertical distribution, which is also interesting, although much more subtle and textured. This is the annual mean depth gradient, by which I mean the value in the top 100 meters divided by the value in the next 100 meters down minus 1. So basically areas that with a 0 on this scale indicate basically no vertical gradient in the metabolic index from 0 to 200 meters. Areas with a negative value here means they're losing, the metabolic index is declining as you go down. And you find that in places where the thermocline is either weak or there's a very strong oxygen minimum zone. Okay, that is, as you're moving down, oxygen is decreasing more quickly than temperature does in a metabolic sense. Okay. The red areas are the opposite of that. They're where temperature is decreasing more quickly, that is, your metabolic demand for oxygen ought to be decreasing more quickly than the availability of oxygen measured by its partial pressure. And in both cases, you know, from the top to 200 meters, top of the ocean to 200 meters, you see something like a 50% variation in the metabolic index. It can be positive, it can be negative. But the point is that it's not a 10 or 20-fold variation like it is across latitude. So, and it's also, you know, it's not a systematic variation. It depends on where you are. So for that reason, we're just going to focus on how the metabolic index constrains the latitudinal range of species habitats. So here's our first example, and it's Atlantic cod, for those who recognize this thing right off the bat. What we're going to do in each case is we're going to take the parameters that come from the laboratory experiments, basically the A0 and the E0, and we're going to use those together with climatological temperature and oxygen to map what that species metabolic index should be. Okay? And that's what's shown in the map in colors. It's a little faint, but 0 down here in black, if there was any, up to 10 in white. On top of that, I've taken the current, or kind of, let's say, late 20th century distribution of where cod is actually found in the ocean, just as presence or absence data. And I just put blue dots. Anywhere that cod, you know, comes up in trawls or what have you, is represented as a blue dot. Okay. Now, with those two things, I asked the question: where in cod's current habitat do we find the lowest value of this metabolic index, and what is that value? Okay. So it turns out that, you know, that value, as you can imagine, it depends a little bit. It depends on the range of the fish that you're thinking about. We're always going to take the size of fish at which populations reach sexual maturity. And the assumption there is simply that that's what's going to govern whether a population remains viable. You know, little juveniles, you know, it's nice for them to be able to stay, but they're not the critical determinant of whether the population is viable over the long term. So we look at adults of sexually mature adults, and in that mass range, we find that at the southern boundary of the cod population, in those blue dots, you get a range of the critical metabolic index from 2.8 to 4.3, whatever. Okay. The interesting thing, I think, is that actually that metabolic index kind of follows along the southern boundary. It's the same in the east, as it is in the west, and it basically follows where you find the cod and where you don't. It seems to be a good, if you were to identify one boundary, one quantity of the metabolic index that separates where you find cod from where you don't, you choose that one, and it would have a value, depending on the size of the animal, of around 3, let's say. Okay, so that suggests that in habitats where there's not enough oxygen, let's say, or temperature is high enough that you can't, extract more than something like 2 to 4 times the energy needed for activity for cod, you won't find the cod, which in turn kind of implies that this might be some kind of real operational limit on how far south cod can get. I'll return to that point in a moment once we've seen some other species. But first you can kind of imagine that this thing varies seasonally too. The metabolic index varies seasonally, so do where you actually find species. And cod is a nice example because there's so many observations. We can actually evaluate seasonal variations in the distribution of the population. So first, just a map of the actual amplitude of the seasonal cycle of the metabolic index normalized by its mean value. You see that for the most of the ocean, in the upper ocean, there's not much seasonality. The variance is, the variation is something less than 10% of the mean value. There are some interesting exceptions. The oxygen minimum zones, again, stand out as quite an exception, because when you're at low oxygen, a small change in the concentration has a big effect. And also in places where you have very strong heat exchange with the atmosphere, so large amplitude sea surface temperature variability also stand out in the seasonal variance of the metabolic index. And you'll notice that for example, in the Western Atlantic is where the cod live. So we looked at whether variations in whether cod populations might be actually following the metabolic index over a seasonal cycle. So here's some time aggregated data from NIMS bottom trawls with adult cod. And on the left is the east coast of the United States. On the left, you're looking at fall conditions, 1963 to 2003. And the dots represent the number of cod that were pulled up in a trawl. Little dots, small numbers, big dots, big numbers. And you can see that during fall, there's a few straggling points down here, but something like 41 north is about where you'd want to stop going if you were fishing for cod in the fall. In contrast, if you look at the springtime, you wouldn't call this a writhing mass, but there's obviously lots of cod extending further south during spring. Some of these points are, in the tens of individuals pulled up. Okay, so maybe wherever you want to call it, high 30s latitude, you'll still find cod in the western side of the basin in these western populations. So we can look at the metabolic index and its latitudinal migration over the seasons from climatological data again. And what we find is that critical metabolic index, depends on body mass size, varies with latitude in much the same way. That is, in fall it reaches about 41 degrees north along this boundary over here, and in the spring it goes down to 37, 38 degrees north, which is where the cod are during the spring. Now there's less clear but more anecdotal tagging data on cod migrations in the eastern population from the Celtic Sea, and it says that basically cod undertake a vertical migration from the surface down toward near bottom depths during the fall, and that depth migration matches well with a seasonal fluctuation in the metabolic index. So we think that this adds further support to the idea that it's this oxygen supply to demand ratio is something that's actively pushing fish around in the ocean, at least if cod are like all fish. They're probably not, but we looked at some other ones too. So here's Atlantic rock crab. And I'm going to go more quickly through these now because it gets a little tedious. We don't have nice point-wise location, but papers descriptive accounts of where the rock crab exists over the seasonal cycle in different parts of the North American margin along the East Coast. And we plotted those against the critical metabolic index, both on the shelf, top 150 meters or so, and along the slope, so the continental slope. So what you're looking at here, it's a little bit hard to envision, but you're looking at the the side of the North American continent facing east, okay, versus latitude and depth, you're looking at the water that lies along the bottom of the ocean. Okay? So we're just following bottom waters and looking at it perpendicularly. And so you see that typical pattern, right, that phi is low in the lower latitudes. Unfortunately, I've lost the scale here. And it goes high toward the high latitudes with a greater amplitude near the surface than at depth. And there's a seasonal variation in the along the shelf, just seasonal heating and cooling, oxygen production and consumption, and it leads to a variation in phi across latitude from the winter to summer that spans the same latitudinal range as where the seasons are found seasonally. Sorry, the populations are found seasonally. So if you're in this latitude range, you'll find these crabs during the winter, but not in the summer because they migrate up to here. Okay. If you're north of there, then you'll find them all year round. Okay. And that's indeed where the metabolic index never falls below this particular range of values. Similarly, if you're on the slope, what people find is that there are annual populations that will persist down to something like almost 25 degrees of latitude. And it's thought that they'll occasionally, that this provides kind of a refuge for some of these migrating populations. And that same metabolic index can be found down to the depth range at which the crabs are observed, down to 25 degrees latitude. So the alignment of the seasonal and depth variations in the population abundance seem again to correspond with the distribution of the metabolic index. Okay, two more. The sea bream, which is interesting because it's more of a subtropical species. It basically runs out. You stop finding it in West Africa, and it has a range of critical metabolic index from 3 to 5 or so, again, depending on the size of the organism, a little bit on the seasonality, but we don't have good seasonal population information for this species. And then a distinctly subpolar, cold water type species, the eel pout, which was the one that we started with in the very beginning from Hans Portner's data, is a shallow, typically kind of deep dwelling down to 40 meters. Well, it sits near the bottom, typically around 40 meters, and its presence data is shown here in blue, and it stops appearing when its critical metabolic index reaches something like, again, 3 to 4 or 5 or so. All right. So that's as many species as we have the data to look at. If we had more, we'd be quite happy. But we can try to draw some conclusions just from the comparison among those species. So going back to this idea from the terrestrial ecology that the actual rate of energy consumption or expenditure by organisms living out active lives of foraging and escape and all the rest of it, imposing an energy need of something like 2 to 5 times the resting rate is exactly the same range that we find when we look at all the marine species that we have. That is that at this southern boundary where the oxygen supply to demand is at its lowest, it gets down only to something like the ratios that you would expect an organism to need in order to have a metabolically viable habitat. So from that, if we had seen, for example, southern boundaries with a metabolic index of 100, we'd say, okay, well, that's maybe interesting, a lot of hard work for nothing, because why would an organism need 100 times the energy that it needs in a resting state? But the fact that these numbers are kind of fairly ubiquitous in terrestrial organisms suggests that there's a fairly fundamental requirement for activity relative to your resting metabolism that ultimately constrains the southern boundary of these species' habitats. So now we can think about climate change, which is where I started with. And in a very abstract sense, we can think about a sample of seawater that's got some oxygen and some temperature, and we begin to warm up the water. Let's say we warm it up by 3 degrees, Oxygen will decline by a certain amount. I think Steve can remind me, 5 degrees per, 5 micromoles per degree, and that drops oxygen by some amount, right? Just pure chemistry will tell us that. And you get some drop in the metabolic index, right? We're on this log scale down here from the temperature effect and some from the oxygen effect. But of course, the ocean is more complicated than that. It's losing oxygen for other reasons that are that get somewhat complicated, but it basically spans this range. You'll lose an amount of oxygen that would typically be that simple chemical prediction, that arrow, but it might be much, much more. You could be losing much more oxygen in certain parts of the ocean because of a whole bunch of other things that are interesting but too far afield. So what I'm going to do then is just use the climate projections for the future to make some estimate of how much the metabolic index we could expect it to decrease over time by the end of this century, just taking averages across a whole bunch of models and a whole bunch of years and a whole bunch of realizations of each model. This is an ensemble mean average. And what you get in terms of temperature is pretty much a global warming signal, right? By the time we're out here, we're clearly outside of the realm of natural variability in the ocean, and this is the model mean warming signal. A lot of interesting structure in there, but it's basically warming everywhere except perhaps where you've got very strong buffering of the warming by exchange with the cold abyssal ocean. The oxygen also has a lot of interesting detail, but on a global scale you could say, yeah, it's deoxygenating, it's losing oxygen at something like tens of micromoles over this century. There are interesting retrograde areas like the tropics, but on average, this kind of pattern seems to be, it's the average of what the models say. We're just going to take it at face value and make some estimates of how big the effect on habitat might be. So first of all, we're going to put those together and just say how much does the metabolic index change. So 0 is no change here in the future relative to contemporary ocean, down to minus 50%. And on a global mean basis, the metabolic index, that is the expected supply to demand ratio for oxygen among organisms, decreases by about 20%. But if you're in some of these high latitude places where the oxygen loss is great and the warming can be relatively rapid, you're looking at something like, in certain areas, 50% loss in the metabolic index, but over relatively large areas like our own North Pacific, 40% reduction in the metabolic index. It's also interesting to think about, since this is kind of a talk about how oxygen constrains marine habitat, it's interesting to think about what oxygen's role in that reduction in metabolic index is. And this was kind of surprising to me a little bit. That even though this is a measure of oxygen supply to demand, it's the oxygen loss itself accounts for actually not even half of the decline in the metabolic index. The increasing tendency of the ocean to provoke hypoxic responses among species is much more strongly associated with this warming pattern. So in these two cases, what I've done is I've just held the temperature constant and let the oxygen change into the future. And in this case, I've done the opposite. And it's the same scale, the decline in metabolic index from the oxygen effect versus the temperature effect. And on a global mean basis, the temperature effect wins out. You can kind of see that. But it's also interesting that there are certain places where the oxygen effect wins out, or at least is highly competitive with the temperature effect. And in particular, the North Pacific, where oxygen starts off already low, and it appears that most models predict a stronger tendency to deoxygenate the ocean. The oxygen actually accounts for a larger fraction of the reduction in the metabolic index. So hypoxia may really be an oxygen story out here, but even if we don't know anything about the oxygen story, we can still think about the loss of aerobic scope in the world's ocean just from warming alone. Now we can look at where these species actually live and just do a kind of mundane, straightforward calculation of how much the current existing habitat gets compressed by this reduction in the metabolic index. Just asking for a given threshold phi value that we diagnose from its current distribution, how much water would start off above that value but fall below it by the end of the century. And just take that volume of water and divide by how much water they live in now. And you get these kind of numbers. And they're not too surprising. You know, they're basically comparable to the reduction in the metabolic index itself. So if the metabolic index goes down by 20%, you're pretty safe with a first order guess that habitat compression will be about 20%, let's say. Okay, so to me this all makes a case for using this physiological data and mapping it to get an actual ecological threshold to make some semi-mechanistic predictions about habitat compression. But of course, as my biological collaborators love to always remind me, the ocean is complex, and there's lots of phenomena that will not be well predicted by just thinking about how the metabolic index changes. So here's an interesting example that's gotten a lot of attention in recent years. It's the Humboldt squid, which has expanded its range north from the Mexican margin. I don't know where it is today, but this is a rather ominous expansion, right? At the same time that oxygen in the California current has been declining, right? So the metabolic index would clearly not predict that kind of behavior. And so there's got to be something else beyond just this either a physiological or even ecological threshold for the individual organism happening. There's probably some kind of species interaction, and people have suggested that the compression of habitat for the prey of the Humboldt squid. So just to conclude very briefly, I'm hoping to convince you that there's a lot of promise in extracting old-fashioned laboratory physiology data from all the millions of file cabinets that still contain this stuff around the world, and using it to extract parameters that allow us to map the metabolic index, the ratio of oxygen supply to demand for different species, because once we have that, we can look at a wider taxonomic variation. For example, we could go look at whether these ideas hold for species that are well adapted to low oxygen environments. You can notice that these are all Atlantic species, not exactly known for its hypoxic tendencies. The Pacific Ocean might offer some interesting contrast. In any case, these species that we did look at, despite being all Atlantic and occupying a range of latitude, they had different thermal and hypoxic tolerances, but when you looked at their ecological threshold, this critical metabolic index that they need to have a viable habitat, they were actually fairly similar. And then finally, given that, we could make projections about what habitat is likely to do, and the surprising point, I guess, it's not surprising that habitat shrinks as a result of warming and deoxygenation, but the fact that it's actually mostly related to warming rather than deoxygenation was a bit of a surprise for me. So with that, I'd be happy to take your questions in here or over beer if the people who left in the beginning didn't drink at all.
So, does aerobic scope vary at all in temperature? I know a lot of those laboratory settings, they measure resting metabolic rate at a controlled temperature, and aerobic scope at the same temperature. But if you're really cold and resting metabolic rate goes down just as a function of temperature, you still need a lot of energy to actually move and do things. So I can imagine this situation where your aerobic scope might need to be larger to be ecologically relevant for a polar organism or a tropical organism. But I don't know if I'm seeing that.
Oh, I see what you're saying. So, in other words, you're saying that latitudinal gradient, for example, in what I sometimes call metabolic scope, this metabolic index, could be less severe of a gradient because actually the ratio of activity to resting metabolism is lower in the cold.
I don't know which direction, but I was actually thinking that the polar organisms might need a greater aerobic scope to be
sounded like a sensible possibility, but I couldn't evaluate it with this data, I don't think. Or at least it's not immediately obvious to me how I evaluated this data. A lot, the physiological literature typically reports metabolic scope as a difference between a maximum metabolic rate and a resting rate. And so it's, if you only have a difference, it's actually hard to relate to this kind of non-dimensional measure of metabolic ratios.
There are physical geographer asked a question.
Yes.
Your time span for cod was interesting because it went back to the period of the extreme warming of the 1930s in the high latitudes. At that time, cod expanded their domain into the Labrador Sea, and the annual yield was something like 600,000 tons of codfish. And this year, they were associated with warming, and I've heard complicated explanations about where they spawn near Iceland and the survival into the Labrador Sea. But how do these extreme changes, which also have clear connections with predators like human fishermen, how do you unscramble those changes?
Yeah. I would love to actually break out that cod trawl data by year and look at whether we can predict, variations in their distribution from even just the temperature alone, think about oxygen variability, in the same way that we do for the seasonal cycle. But I do have this sense, I share the sense that the human imprint could be confounding. And I think that's why, that paper on the Southern California mesopelagic fish that are not big commercially fished species suggests that, or at least not all of them, suggests that there could be a detectable oxygen temperature effect in these things, but cod will be a difficult nut to crack. Many people have tried that.
And the disappearance of cod in the 1970s, 80s, and 80s, which was monumental, and of course, endless argument about cod and climate versus cod and overfishing.
Yeah, I haven't looked at either one. Just taking, both time being kind of picture, but I think that would be really interesting to do with someone who actually knows the cod data better.
Yeah, I see.
Yeah. Oh, there's tons of people who could do it. Yeah. Anyone in the room done? Oh, Steve's done it.
No. So if there's it's pretty simple. There's temperature variation for fish, and things are getting warmer, then warmer fish will move colder habitats. Does that mean that the only losers are really the ones that are in the coldest habitat because they get fish moving into their range?
Yeah.
I'm not really sure why it has an effect other than just moving things around.
Yeah, so I would not use this kind of reasoning to make predictions about poleward expansion, simply because at the poleward range limit, the scope for metabolic activity seems to be plenty high, and decreasing the metabolic index from 20 to 15 ought not to be stressful. So I think that the reasons for northward migration at the poleward limit, for example, are probably more to do with things that dictate the tolerance to coldness that don't have anything to do with oxygen. That's my sense of it. But there's interesting, one of the interesting things about this that, to me, is that people have looked at range limit shifts in marine species at the equatorward side and the poleward side, and they find that the fraction of equatorward side contractions is greater for marine species than it is for terrestrial species, where your intuition about warming, making something better, more polar, actually does hold up. So I can maybe make more sense of that over the beer. But I think that this metabolic reasoning should really be applied at not so much at the polar range limit. Hillary.
So I thought you made a pretty strong case that this metabolic constraint sort of aligns with the southern limit of where these species are found. You're sort of assuming here that there aren't any other factors affecting where these species might be, like things they're eating or things that might be eating them or the rest of ecological interactions. So I'm wondering how you can evaluate those alternative hypotheses to sort of show that it's this metabolic constraint rather than perhaps other things that can control the species rate.
Yeah. So cod is probably a good example. The metabolic index would say that from an oxygen supply to demand point of view, anywhere north of that black line should be perfectly viable habitat, right? And so there's two parts to your question. One is, well, why is there perfectly viable habitat that's not being exploited? And the answer clearly is something besides metabolic index, right? It's probably availability of food or something else. Whether the metabolic index. So the other part of your question, if I've understood it right, would be that how do we know there isn't actually something else that's keeping the cod at bay at these latitudes, and the metabolic index is just some coincidental number that happens to line up with that true thing, some ocean monster that's pushing them back. And I can't say that there isn't. It just strikes me as too much of a coincidence. If I had seen a metabolic index at that southern boundary of some ridiculous number, like 50, or I would say, well, it's interesting, it lines up, but it's not. It can't be the operative constraint. There's got to be something else. But the kinds of numbers we get, just I don't really see how cod. To put it another way, if there is something else operating there and you took them away, I still don't see how cod could really migrate much further, unless you're willing to admit that cod violate all the rules of all the terrestrial taxa, and they really don't need that much energy to live. Does that make sense?
Yes, I would want to know what are the cod eating and what's their metabolic index, for instance. Maybe the cod are less sensitive than something else that they're coding, for instance.
Oh yeah, I think when we get into how a change in metabolic index will split up and realign communities, all those, we really need to know the constraints on interacting species. No doubt about it. That would be the Humboldt squid example. I don't know, how far away that is, because I'm not going to put fish in a tank myself.
I think something to add to that is that there are people who study the ecosystem dynamics relating to all these different species, I'm sure, but particularly Atlantic cod, but they ignore metabolic rates. So probably just an opportunity to work with other people.
Yeah. I mean, I've looked at some of that stuff, and usually what I find, I'm actually turning this around as a question to you, is that, cod don't like waters warmer than 11.3 degrees or something. And that seems like a sensible description of nature. But 11.3 to me sounds like kind of an arbitrary number, and I don't know how to make sense of it in any other way. And so then I always want, then I want to, understand why it's 11.3 and not 5 or 20.
Well, I also don't know about physiology. I just think it's interesting that you have the physiologist, and then you have the oceanographers, and then you have your population dynamicists.
Mm-hmm.
And it would be cool to bring that together, just because I see this and I'm like, oh, this is that piece where we don't think about that ever.
Mm-hmm.
And then I also probably don't really think about temperature ever. But we have the models, the population models going.
Yeah. It would be interesting to look at, to compare this with like, their food production.
Yeah, exactly.
Which one
Yeah.
fisheries people do. So,
Exactly.
yeah, I agree.
Not me specifically, but it would be cool.
I agree.
This is really a follow-up on the same line of question. You've got a slide that shows the seasonal variation between spring and fall.
Mhm.
And I know you were looking at north-south, but it looked like the distance east-west the picture was greater. And I'm wondering if that may relate to some dimension of habitat that could be other than temperature or O2, or maybe it's some function of them in a way that is also revealed in the data. I don't know.
Yeah, interesting. I mean, I confess to have, distilled this data into a single number, and there's a lot more going on for sure.
Yeah,
And I hadn't noticed the east-west thing.
It's mostly a westward motion more than it is a southward motion. And I'm looking at that. Is that having to do with some performance of the coastal habitat distinct from O2 and temperature? Or is that a function of the temperature and O2 data that could tease something out here? I just don't know.
Okay, just help me identify what you're keying in on. Is it
If you run to
this
the
kind
right
of
from there,
tail that goes down?
That swarming mass of cod along that western shore.
Yeah.
It's not there
Right.
in the springtime
Right.
or in fall. It is in the spring.
Yeah.
Does that correlate with any of these, with the metabolic index, for example?
Well, I mean,
Or is it a distinct?
I think of it as a southward kind of migration rather than an east-west, because, where are they over here to migrate eastward from? But you're right. I mean, it is kind of a
The distance west is greater than the distance south.
yeah, sure,
I'm
yeah.
thinking, and it's
Yeah.
a
Actually, that-
kind of habitat.
Yeah, I should clarify that latitude thing that I showed you looked at grid cells that followed the coastline. So they were both north-south and east-west. I was just looking only east-west at the grid points. So it included both of those things. But yeah, there's also depth. They go to different depths in different seasons, and that's something that would be interesting to look at. But yeah, if anyone in here is a cod expert and wants to do more with it, I would love to talk to you.
All right, let's give Curtis another round of applause.